what are genes? An analogy I like to use for genes is they are like a recipe in a cookbook. Genes are a set of instructions for the body on how to make a certain product or how to perform a certain function. Each of us has about 25,000 genes, and we call that the genome, like the cookbook is for the recipe, the genome. And together, all those 25,000 genes pretty much tell us how to live, tells the body what to do, how to function, how to make itself, how to repair itself. Like animals and plants, we are all made up of cells, liver cells, muscle cells, photoreceptor cells, the light-sensitive cells in the eye. And those cells are pretty much semi-autonomous. They can live by themselves, and that's because every single cell has a copy of the genome. It has a copy of all those 25,000 genes inside it, and therefore it can live by itself. They don't necessarily use all those cells. So a liver cell, for instance, will use the gene that tells it how to degrade alcohol, but it won't use the gene that tells it how to sense light, the rhodopsin gene, for instance. The rhodopsin gene is only used by the photoreceptor cells because they are the cells that actually have to make the light-sensing machinery. So if there's a mutation in a gene, if there's an error in that instructions, like leaving out a certain ingredient in a recipe, it goes wrong. The product that the gene should be making can't be made, or the task it should be performing can't be performed. And as a result, the cell that uses the gene can't function normally, and there's a disease, usually a disease as a result. Now, what then is gene therapy? Gene therapy as a concept is very, very simple, really. If we know that a certain disease is caused by the fact that there is a mutation, an error in gene X, then if we can actually put a normal copy, a functioning copy of gene X back into the cells, then basically the disease should be cured. We should have made the cell normal again, and therefore it should function normally again. We could try to put that gene back in all the cells in the body, but there's about 40 trillion cells in the human body, so that's not going to be, it's not going to work, to be honest. But of course we don't need to, because as I said, not all the cells use all the genes. So we only need to put the cells back into the genes back into the cells that actually need that gene. And in the retina is fortunately quite small and doesn't contain that many cells, so it's actually quite a feasible thing to do in the retina. So how do we do that? How do we put genes back into cells? Well, nature has come up with a solution for us. Over millions of years, viruses have evolved with only one goal in mind, and that's to put their own genes into the cells of the host organism. And we can hijack that mechanism. We can actually take virus particles, scoop out the viral genes, and put back the genes that we want, that we are interested in, the genes that we want to put into the cells. And when we then inject those viruses close to the cells we're interested in, the virus will then put that gene into the cells and hopefully cure the disease as a result. And in the eye, there's been a lot of research in gene transfers, we call it, putting genes into cells in the eye. And there's one virus which is particularly good at it, and it's called adeno-associated virus, or AAV. And we're lucky, not only is it very good at it, but also AAV doesn't cause any disease normally. So it's not only effective, but it's also really quite safe to do. So what has been achieved in gene therapy? Well, gene therapy has been going for 20, 25 years or so. And I'll be honest, the first 15 years were slow going. As I said, there was lots of research in how to get genes into the eye. The concept was simple, but actually doing it wasn't all that simple. What genes do we need? How do we actually drive those genes, etc.? And over the first 15 years, slow progress being made. AAV was discovered. And finally, in about 2008, I think quite a few of you will know already that about 2008, we were the first group to do a gene therapy study in clinical trial in patients here in the UK. It was for a disease called Leber Congenital Amaurosis Type 2. It's caused by a gene called RP65. And basically, patients with that disease don't have night vision or very little night vision from birth, and they have a progressive loss of their daylight vision over time until total blindness by the early 20s, approximately. So we made a virus, an AAV virus, or AAV vector. We like to call them vectors because it sounds better than viruses. An AAV vector that contains the human RP65 gene. We injected underneath the retina, as close to the photoreceptor cells as they could go. And then basically we looked at what does it do, what's happening in those patients. And we could show that there was a recovery of the night blind. So there was an improvement in night vision. We could show that these patients, they could walk through a maze under dim light conditions, whereas they previously could not do that. We could measure the light sensitivity of the retina, and we could show there was better light sensitivity to very low levels of light. So basically, we could show that gene therapy can work. The concept of gene therapy is right. In the retina, we can put a gene back in the place it needs to be, and we can then restore some of the function of the retina, of the cells in the retina at that place. However, and there's usually a however in medical science, we could also show that the function of the retina wasn't completely normal. There was an improvement, but not a complete improvement. We could show, when we followed those patients over several years, that the loss of vision over time was still going on. And basically, although the treatment worked, we could show an improvement. We could get the gene in the right place. It wasn't complete. And we concluded, and we were not the only group doing this type of research, two other groups in America, and one of the groups concurred with us that although the treatment showed feasibility, it was not perfect yet. It needed to improve. Of course, we didn't sit still. The scientists didn't sit still when the medics were doing the treatment of patients. Of course, we continued our research. We looked for better ways to do it. And then I'm talking about the whole gene therapy community worldwide. We looked at better vectors. People discovered new strains of AAV which were more potent, more effective than the strain we were using before. Some groups even designed new strains themselves with specific characteristics. We looked at ways of making the genes more potent once they had infected the cells, make them more effective, give a better treatment effect once the infection had occurred. And taking all those improvements together, we could test them in the animals and could show that we could now treat animal models of retinal degeneration, mice and dogs, and with retinal degeneration, which previously we couldn't, very severe forms, we could now treat with these optimized vectors. And the result of all that is that gene therapy now stands at the cusp of a big boom, the retinal gene therapy. We know it's feasible. We've shown it before that the idea is feasible, and now we are getting to a point where we can really start to use it. We ourselves, a group, we've optimized the RP65 gene therapy, the old vector. We used a new strain of AAV. We used a better, stronger gene to put in there, and our new vector is about 300-fold more effective, more potent than the old vector was before. And whereas with the previous one, we could show an effect and get a bit of an improvement. Hopefully now, when we use this, we can get a real strong, complete normalization of the retina. We've received funding from the Medical Research Council to start a clinical trial with that vector. Hopefully, probably in the spring of 2016, so in only half a year away or so. And we hope that that vector will be the real evidence that retinal gene therapy is practical, but it can also provide a lasting benefit to the patients. We also have developed and optimized a vector for a disease called Achromatopsia. It's called by a gene CNGB3, and that's a disease in which patients have defective day vision. So these patients don't see color, they don't see detailed reading vision. And again, we have received funding from the MRC to start a clinical trial for that disease with an AAV vector and CNGP3 transgene. And we intend that trial to start in the autumn of 2016. So again, within, well, around a year from now, we expect to have 2 trials running for retinal degeneration. Even more excitingly, I find, is that very recently we published a paper in which we showed in mice that we could treat Xlinked RP3, a gene called RPGR, and that is actually the most common form of inherited retinal degeneration. And we can treat that disease in mice, and although it's a bit not as far evolved yet, so I can't give you a precise time frame, but we are working with commercial entities to help us get the money together to start a trial for Xlinked RP3, hopefully in the next 2 or 3 years. And that's, if we could treat the most common form of inherited retinal degeneration, that would be a real step forward. We're not the only group working on gene therapy for retinal disease. We've already heard this morning that there's a trial ongoing for Stargardt disease, ABCA4 gene. I know for several other trials in the US, in Europe, and although we know there's 200 different genes and they all have to have their own gene therapy developed, we are really starting to see an increase, and I'm sure that over the next 5 years there will be 10 different gene therapy trials probably going on everywhere in the world for different genes. And I think we can really start to see a change in not just the treatment, but also the attitude towards inherited retinal degeneration as an untreatable disease. That leads me to thank basically not just the MRC. I've mentioned the MRC as a sponsor already a few times, but I also should mention RP Fighting Blindness, who has been really helpful in our research, sponsoring our research for the last few years. Thank you.
Do you really mean regeneration of lost sites?
For this particular disease, RP65 disease, and also for achromatopsia, it's basically lost sites. The cells are there, but they're not working. So once we get the gene in there, they will start to work, and site that wasn't there before will come back or will appear. That's not the case for all diseases. So there are also forms of disease, for instance, RPGR, where the site is slowly lost, and once it's lost, it can't come back. So there we would expect to see a stop of loss of sight, right. Hopefully.
Yeah, thank you.
Thank you. 2 related questions. The first one is, do you know why the current treatment is not as effective as you might have hoped? Is it a lack of penetration into all of the cells, or is it a weakness in the gene? Or
basically, our idea, and not all the research groups agree on that, but our idea is that as I said, genes describe how to make a product. They actually make the products themselves, and we think that we don't make enough product. So we have got the gene a vector which drives 300-fold more RP65 protein production, and we think that therefore we can get the retinoid cycle going really properly again.
Okay, and I can understand what you said about producing a more powerful vector to spread more virulently. What I don't understand is the term stronger gene. If you could explain that a little.
Yeah, so as I said, the gene itself, it's not just an instruction, it's also the actual process of making the product. And so by having a stronger gene, you just end up with more of the end product from that instruction, and therefore with more protein or more product made, more molecules made, you basically get a bigger treatment effect, usually.
Yeah. Can I just ask why you chose CNGB3? Is it because it was the most popular form of the genetic disorder?
There's two main forms for this disorder, CNGA3 and CNGB3. They are approximately equally common. We chose B3 because we know other people are working on A3, And so, but then again, there's also another group working on B3. So it's spreading the load a bit. There's a German group about to start a CNG A3 trial, and we don't want to double up the research too much, I suppose.
Because I was also thinking, if we take it to the analogy of like your recipe book, do you actually, I mean, you put yeast in a recipe and it makes the bread rise. Do you actually know what each gene's participating function is or not?
For, I'd say 95% of the genes we do, not all of them. There's a few diseases where we just go in semi-blind. We know it's needed, but we don't really know what it does. For CNG A3 and B3, for instance, we do know. I'm going to get very technical now. They're ion channels. They basically generate the electrical signal that goes to the brain.
Thank you.
What makes you think the brain will then react like it should?
Yes, that's a very good question. We know from children who have a lazy eye that the brain at some point loses its ability to react to a signal which has never been there before. And so in older patients, we don't know. We hope. There are some indications that actually, even in adults, 25, 30-year-olds, there is still an ability to learn in the brain to new stimuli, but slower, less effect. So ideally, we want to go younger and younger until we reach a point where basically the brain is still fully receptive to new stimulus.